from Microbe TV. This is Office Hours. I'm Vincent Racaniello, and welcome everyone to another episode. Well, this is the premiere episode, right? It is the premiere of a new live stream that I'll be doing, mostly on my own, but sometimes with guests. I'm Vincent Racaniello, and for those of you who don't know me, I'm a professor at Columbia University. I study viruses. I have studied viruses for over 40 years. I have written a textbook now in its 5th edition on viruses. I have done podcasts on viruses. I've done a lot of virus stuff. I know a little bit, don't know everything. And I thought I'd answer your questions. Office Hours, of course, is the name for what I do with my students in my virology course, which is currently running at Columbia. I have office hours once a week where they answer their questions. So I thought I would do the same thing since the lectures, the virology lectures, are being posted on YouTube. I thought I'd do the same thing for all of you out there in the world to ask your questions about viruses. And so welcome. Hopefully this will go for a long time. I'm planning to do it once a week, 8 p.m. Eastern Time U.S. for about 2 hours. And once a month, though, we will go back to Q&A with A and V. I think I would love to have Amy's voice with mine. She's a great resource, and we'll do that once a month. And altogether, every week you can get your virology fix. A couple of items of business before we get started. So we have moderators here, of course, in order to keep the discourse civil. And let me welcome our moderators who were previously on Q&A. We have Les, we have Frank, we have Steph, and we have Tom. Welcome back, and thank you for coming. And then we have two new moderators, Barb Mack from the UK, and PDK, Peak Dunning-Kruger. So they're all. Thank you for joining us here, by the way. They're here. They have the blue names in the chat, and they have a little wrench next to their names so they can fix things in case they break. And so thank you all for coming. And the YouTube channel where you can find the lectures, let me show that. I believe I pulled it up here. Here we go. So all of you, many of you know the YouTube channel. It has a new look, by the way. We're rebranding. It has a new header. There's a new picture of me. And here you can find my lectures. So there are 4 so far that have been posted. And they are free, of course, for you to peruse. And if you have questions, you can just bring them here. So what kind of questions am I going to take? Most viruses I should be able to handle. If I can't, I'll tell you. I won't give you medical advice. I'm not a doctor. I'm not an MD. I'm a PhD, so I can tell you about the basic science behind it. And those are the kinds of questions. And if I don't know the answer, I'll ask Amy, who will probably be here. All right, and we'll do that. Let's see, what else? Oh, this is very important. So we have a Discord channel for Microbe TV. And I invite you to go and hit that link and join the Discord server. It's free. It's a place to chat for all people who are interested in the subjects of Microbe TV podcasts. And there are nice places where you can go to talk about all the different subjects. We have a wonderful administrator, Mr. Mason there. PDK is actually a mod over there as well. And we have over 400 some members already. So pretty exciting place. I like to spend time there, and so you should go and you can ask your virus questions. You can have this community here 24/7 if you'd like. I will leave that up for a while. So as I said, tonight we would usually go for 2 hours. Tonight I'm going to stop at 1:15 because at 10 I have to record Daniel Griffin's TWIV clinical update. He's somewhere else and he has to record today, and we could only do it after this stream, so we'll stop 15 minutes early. But in the future, we can. Every now and then, I'll surprise you with an after hours. So I have to let the mods know ahead of time, but I will surprise you once a month or so with an after hours that'll keep going. Office Hours After Dark. All right, let's see where people are from. I love to find out where people are from. By the way, tomorrow we're going to do a TWIV special with Paul Offit to talk about the latest FDA Advisory Board meeting. So that should be fun. So Armand Franke here is in New York. Let's see where other people. Barb Mack, of course, is from the UK. Let us see. I know Steph is from San Francisco. Nicola is from Italy. I know Quistela is from Ohio. I already know that. I remember. Some people who frequent often. I remember. Genetics 2 is from Arizona. That's cool. Let's see, who else? First, yes, this is the first episode. It is the premiere. Oh, maybe not the last, okay? There are already 220 people, so it's already a success. And many of you were on Q&A. It's the same crowd. I know Tom is from Oregon. There you go. Maureen is from Ohio. Richard is from Santa Barbara. Let's see. Hamilton, Ontario is Monica. Circus Pup is from the California Bay Area. I have been on a few cruises. What's going on? This is not the new Q&A. This is Office Hours. Q&A will be once a month. It's okay. Virginia. Jem is from Virginia. Cool. The professor is in. Ooh, I like those books. Very cool. I'm a professor. I teach virology. And yes, I miss Amy too. We will have her back later this month. If she's listening, we miss you, Amy. Italy. Paul is in Italy. Very cool. Love it. I will have guests. Yes, for sure. I'm planning to ask Daniel first and a few others. So stay tuned. A lot of guests. Okay. So no more people. Oh, yes. Here, Hillsborough, North Carolina. Welcome. John. Hey, we've met. Nice to see you. We met in Connecticut first. Windsor, Ontario. Oh, now they're coming in. Yeah. Eastern North Carolina. I'm going to take time doing this because I like to know where you're from. I'm sorry if some of you don't like it. Lisa is from Oswego. Yes. Tracy's from Staten Island. Susan is from New York. Ariel is from Westchester. Dawn is from the Netherlands. Cool. Bettina is in Munster, in Indiana. My Laughing Hog is from Arkansas. You know, there's an app I can get that will display a globe with all of your locations. I should get that so I take less time. Here we have North Carolina, Columbus, Virginia, North Carolina, Vancouver, BC, Illinois. Here's Rafael from Brazil. California. PDK's in California. Thanks. I didn't know that. Pittsburgh, PA. Denver. Matthews, North Carolina. Another North Carolina. Yeah. Stacy, I think we had is Ontario. Lowell, Massachusetts. Lexington, Kentucky. Singapore. All right. I love it. Oh, Vanity shows up here. Yes, Vanity is here. I'm sorry I didn't mention you. Another one of our moderators. I didn't see your name there. Yes, I will get Brianne on as a guest for sure. South Carolina, Vancouver, BC, Leawood, Kansas, New Orleans. You know, I'm going to be in New Orleans in April, I think. Sydney, Australia. 67th and 3rd. Cool. I'm not far from you. Now I'm in New Jersey, but the incubator's on the other side of town, but it's not far. Boston, India. Cool. LA County. Eastern Massachusetts. Minnesota. Arlington, Texas. Houston, Texas. where I will be this summer. Another from India. Very cool. Seychelles, BC. And here is Doreen from Illinois. Here is Chicago, Northwest PA, Tennessee. You don't like to be called I'll call you Monique. Why would I call you Monica? I don't like Vince. No, you want to call me Vincent or Vinny is fine, but not Vince. Hello, Eric from Boston. Nice to see you. Winona. Very cool. Right. A couple more here. Newfoundland, Rhode Island, Columbus, Ohio. Okay. That's enough for now. Costa Rica. We can't spend the whole time just saying where people are from. There's going to be like 400 of you, right? So let's go back to the top and do some questions. I'm going to take easy ones first. Okay, you got the Discord server. Can I take that down now? Okay. I'm going to take easy ones to get me going. You know, I when I do interviews with people, I always ask them, tell me your history. It's an easy bunch of questions. Anybody should be able to do it, right? And it gets them at ease. And then there's like, oh, this is easy. And then you start the hard stuff, right? Oh, yes, I said Paul Offit will be on TWIV Special tomorrow to talk about the FDA meeting. And I hope to get it up Friday, but I don't know if I can do it. I would like to, but I have a lot of other things to do. Okay. I listened to the recent Viroids show. Explain how a bacteria viroid enters and then replicates. I don't know what you mean by so you're talking about the paper we did, yeah, where they found a lot of viroids that are apparently from bacteria, right? Well, it's a good question because all we know are the viroids that infect plants, right? And they get in when the plant cell wall is damaged. Either that or an insect will deliver them. And then, depending on the kind of viroid, they're copied by enzymes in the cell to make little viroids that will then leave the cell. So we don't know how it would work in bacteria. We wouldn't know how they would get in. They could, there's not going to be any damage that happens to bacteria. There's not going to be an insect that injects a viroid into a bacteria, right? Maybe they're brought in by phages. It could be. Well, bacteria do take up nucleic acid, so maybe they could take up the RNA as well. And then some enzyme has to copy them. And it could be DNA-dependent RNA polymerase or DNA-dependent DNA polymerase. We don't know. Those are the possibilities, and we'll have to see. All right. My first question, thank you, school email, is appropriate. My goal is to teach the world virology. That's why I started Microbe TV. That's why I started. I just started podcasting on a whim, but Microbe TV was founded many years later, and it's to unify all of this education that we're doing. As many people as possible, and supported by your philanthropy. So if you want to support us, you can go to microbe.tv. Let's see, where is the support? It's Microbe TV Inc. That's not what I want. The Amy Papers is not what I want. I need a better thing here, huh? Well, it's microbe.tv/contribute. You can go there. Excuse me. You can go there and donate, or you can go here. There should be a donate button below the video right here. It's our mission to teach the world about all kinds of microbes. I do the virus part. Frank wants to know, is it possible to get flu twice in a month? So if you have a great immune system, or even just a good immune system, you shouldn't get it twice. So, the boost in antibodies and T cells that you get after the infection should protect you for at least a couple of months from another infection. And I'm guessing you're thinking symptomatic because I say you're going to pick it up. However, if your immune system is defective, then you will be able to get it twice in a month. So it really depends. And you may not know that your immune system is defective. So I would say yes, but the frequency is going to be low in the general population. I heard an interesting factoid the other day, and here's one thing you're going to get from me is I'm going to draw on the stuff I hear on a day-to-day basis and tell you. So I was at NIH last week giving a seminar, and I met with John Udell. I met with him for like 2.5 hours. We talked the blue streak. Well, I guess mostly he talked the blue streak. And he said, the most common immunodeficiency is secretory IgA, right? That IgA, which is supposed to protect us. And those people don't get any more respiratory or mucosal infections than anyone else. So go figure. So I'm guessing that if you had secretory IgA, it would help. But if you don't, apparently it doesn't hurt. So there you go. There you go. Okie dokie. Let's find another question. We have to go down the list here. Everybody's all excited in the beginning. Here we go. Nicola said he's finished, or she is finished. In Italy, Nicola is a guy's name, right? And Nicole would be the girl's name? I don't know. I'm going to put my foot in my mouth. Anyway. I finished Oshinsky's book on the history of polio and loved it. You have another book on polio to recommend. So the one I did recommend for him is The Cutter Incident by Paul Offit, which will tell you the history of litigation, product litigation in the U.S., really, because that first Salk vaccine released was defective. It wasn't completely inactivated, and so a few hundred kids got polio, and so the lawyers came out of the woodwork to start suing, and it was a big, very, very public trial of a young girl who had gotten paralyzed. Her parents sued the manufacturer, and so it's a good story, all about that. But thank you, Nicola, your mail. I'm sorry. It's a good story that talks all about the development of the vaccine and its problems. Now, what's interesting is that when I interviewed Julie Youngner a number of years ago, so Julie Youngner worked with Jonas Salk, and in his interview, which you can find on Twiv, he said he went to, so Salk sent him to visit the Cutter Labs, which was a California laboratory, one of several making the Salk vaccine. They now make insect repellent because I guess they wanted to get out of the vaccine business. I don't blame them because they made a mess of it. But Youngner said, I saw the procedure and it wasn't right. And I told Jonas, you have to do something about this or there are going to be problems. And he said, Jonas said, yeah, I don't have time. And he didn't do anything about it. And he thinks that's why there was this Cutter incident, because they didn't listen to him. I don't know if that's true or not, but that's what he told me. It's on the episode. He said it on the episode. So it's not like I am revealing anything that should be kept hidden. And then Peake in response said, Jonas Salk, A Life by Charlotte de Croze Jacobs is a pretty solid book about Salk also. So those are two there. And there are others, but you can... Splendid Solution is also about Salk. That was by Jeffrey Kluger, I think. Yeah, Jeffrey Kluger. Yep. Okay, next question. You see the questions are few and far between because everyone's excited to be here. 300 people. This is cool. It's more than I expected. I thought there'd be 5 people and then we'd call it a day. But I'm glad you're all here. Simon is in San Jose, where they're decommissioning the mass vaccination and testing sites. Well, that reminds me, I forgot to do something. I was gonna do some Amy papers, but I think I'll wait because there are plenty of questions here. I'll save them for next week. I'm just looking for some questions. Okay, the viroid question there is repeated. A History of Poliomyelitis by John Paul is yes, it's an excellent book, but it's out of print. You're going to have to spend $400 on it. Yeah, I actually bought 2 copies at high price because I think it's a great book. It doesn't actually go past the Salk vaccine. It doesn't do the Sabin, but it is nevertheless good. And I have some others. I don't think I have any here. But when I get to the incubator, here's the Cutter incident for example. This is really good. How America's first polio vaccine led to the growing vaccine crisis. Really good. So read that first, Nicola. Now, Nicola was the name of my first graduate student, Nicola LaMonica, who was also from Italy. You bet. And hit the like button, folks. 300 of you, 191 likes. Let's get it up over 250 so more people will find us. This is a new stream, so not necessarily going to be advertised. Nicks and Mikes are enjoying the discourse in the Discord. Yeah, it's great there. Go visit it. Let's put it back up again here, if I can find it. Discord, there it is. Go join. Even if you think you might not do much there, just go join and pop in every now and then. You're going to see great questions being answered, resources being posted, paper links, and so forth. Really good. And here's an example of a donation. Thank you, guys from San Francisco, Kip and Laura. Thank you for your contribution to science education. Miss Amy and the two of you as the lock horns. Yes, well, we will lock horns later in the month. We certainly will, but thank you for that. I do miss Amy too. Amy, you there? Say hello in the chat. Just everybody will flip out if you say hello. They'll love it. So Vaxman saw my GP talked about Paxlovid. She'll prescribe it if I need it. Very good. That's a plan according to Daniel, for sure. All right. Have you seen about repeated COVID-19 infections causing weakened immune system, which makes you more prone to get sick or have severe disease? So I don't find those reports convincing. I think that may happen in a very few cases, but it is not the rule in repeated infections. It doesn't destroy your immune system. It doesn't weaken them. I haven't seen good evidence for that, no. So I don't think it's contributing to, for example, long COVID. But like everything else, it is always subject to new research. You know, science always changes, right? You say, this is the way it is, and then sometimes later it changes. And some people feel that's an indictment of science, that they should get it right the first time, so therefore they have mistrust in scientists because they say things and change their mind. But in fact, that is how science works, and that's how it should work. Everything you find is not right, or it may be right at the time, but it gets modified as approaches improve or change. So that is the flux of science, and it should not be seen as a negative. And that's one of the things we have to spread the word about out there with our podcasts. Okay, is there any new research to improve the current vaccines? For sure. People are always trying to make new vaccines. People are trying new delivery methods like nasal sprays, microneedle patches. They're trying different antigens, different vectors. This is a virus against which we will be vaccinating forever and ever and ever. So there's always a market for new vaccines. And so companies are always going to be making them. Right now, Pfizer and Moderna are making a lot of money on vaccines, but they're continuing to tweak them, and other companies as well, because there's enough to go around. So don't worry, it's happening. And we'll cover it as it happens on our programs in here. Both RSV and SARS are enveloped RNA viruses that infect respiratory tract. What could be some reasons that fomite transmission is considered significant for RSV but not for SARS? Well, to be transmitted in a droplet that comes out of your respiratory tract requires some physical properties. And so those are different for just a virus sitting on a surface and being picked up. And I don't know what they are. I don't think anybody knows what they are. But that's some of the differences that you have to take into consideration. What is happening, as we know, in China with respect to COVID? I don't know that we can really know what's happening. I think the information is sparse and a lot of hearsay. I did get a link to. I got something from someone describing what's going on who's supposedly there, and I haven't looked at it yet. So I'll take a look, and next week we can expand on this, for example. Okay, Eric, thoughts on ideal time to get the second dose of Shingrix. So, on dosing, the CDC is often correct, not on everything else. A student came up to me before class today. She said, "Professor, I read on the CDC site that they said bathrooms are not dangerous to get virus infections." And I said, well, that's not actually true. Toilets make aerosols, and you could get norovirus that way. Anyway, I think 2 to 6 months is good. I went in, September to get my first Shingrix, and the pharmacist said, come in December, right? September, October, 4 months. And I ended up going in January, which was fine. So I think 2 to 6 months is absolutely fine. I can't tell you the data that would support that, though. That's a good question, and we might want to look at that, but I don't have it in front of me. Can I explain what Pfizer did that the right wing is lying about? I don't know. I've gotten a bunch of emails about this, but I haven't checked it out. I prioritize the science that's published, right? It's hard to get into this sort of thing, but I am planning to look at it, Tess, and we'll let you know. We'll give you an update for sure. Technically, it's already after dark. It is. Should we call it after hours when we go late? Yeah, maybe it's after hours. I'll write that down. After dark has a nice ring to it, though, right? Yeah, at least you like after hours? Okay, good. We'll stick with it. and that happened a long time ago. Okay. Oh, I forgot to tell you that Rach is on the Isle of Man, where they actually have bagels. How about that? Jacksonville, Florida, Silverton, Oregon, Wellington, New Zealand. Cool. Lisa, thank you for your support of science education. Really appreciate it. Hit that like button, folks. Ontario, Canada, Phoenix, Bologna, Brazil, London, West Virginia, Florida, Columbus. I'm just going through the thing here, and I'll get to questions. Sydney. I'm just amazed at the stretch, right? It's really amazing. Elise has been watching the 2023 virology lectures and enjoys them. I've learned so much. You're welcome. And everyone should enjoy them. I'm putting them up there for free for you to enjoy. And it's cool that Columbia lets me do it, right? I think that's great. Pennsylvania, Santa Fe, Seattle, Manhattan. You can come visit the incubator, aren't you? And James got his stickers. So there may be some new people here. If you want one of these microbe TV stickers, just send me an email, vincent@microbe.tv, with your address, your postal address, and I'll send them to you. I have sent out a bunch of these, and there may be some new people here, and you can get them for sure. There's actually a bigger version of this that goes on your car bumper. But you need to be a Patreon supporter to get that, because they're expensive. I'm not being cheap. I just need to support this company, right? Okay, more places, which I think we've gone through. Angela's been listening since 2016. You should come to the incubator. It's a cool place. And we have a great Ebola painting. There's Vanity. Hello, Vanity. Welcome. Sorry I didn't catch you initially. From Virology 2023. Does any type of RNA or DNA reduce incubation time, or is that completely unrelated? I assume you mean if you put viral DNA or RNA into cells directly. yeah, you're going to bypass the entry, but I don't think it's going to make an appreciable difference in the incubation time, because it's just the first round, right? The DNA or RNA is going to get into cells. It's going to make virus in whatever 8 hours. It's going to come out, and then the virus particles are going to infect new cells. So unless you got enough into cells to do a one cycle, it wouldn't really make much of a difference. So yeah, we'll get Brienne. We will get Bill Gates. He wouldn't come on. Somebody has a connection, let me know. I'd love to have him on this live stream, but he wouldn't. And Angela said she will come in and pick up the sticker. Sure, do come in. And some people say, well, I'm a Patreon member. I didn't get a sticker. So I just did that today, but I would be happy to send you a sticker. Just send me your address and I'll send it to you. No problem. How's the planning for the 1000th episode? Well, so the 1000th episode is going to be on April 15th in New York City. I'm trying to get a theater. And Amy had found a few theaters for me, and I emailed the Joyce, which is not far from the incubator, and they're, of course, booked for their dance season. But they gave me some other theaters in Chelsea that I'm looking into. The Joyce is cool because it's 400 seats, and I think we could fill it. And also do some fundraising, right? We could have people donate a certain amount of money, which would be tax deductible, of course, because we're a 501c3. But I'm working on it. And the problem is that, I have other stuff to do, which is really interesting. So what do you think gets pushed to the side? Yeah, doing the theater rental, but I have to do it. Then I have to get a sound person, a light person, a video person, right? Be cool to have a theater with a bar so that people could buy drinks. Oh, and get this. This is a very cool part. So, Dixon's nephew is a saxophone player, and he has a little group. I think he has a trio. And they've agreed to come and play before and after the TWIV, right? So there'll be a little band playing there as you come in, and then they'll stop, and we'll do our TWIV. And then when we say goodbye, the band will play again, right? Kind of like a TV show. It's very exciting. I think it should be cool. So what was I saying? Yeah, that's the 1000th episode. We're going to have it. If we end up getting a 50-seat theater, then we can't accommodate a lot of people. But if I get a bigger theater, I'll let you know. I'm here every weekend. You can come here and find out, right? A lot of people telling me, oh, here we go. This is a question. Lori writes, TWIV episode 604. Dr. Chumakov. So this is Kostya Chumakov, who basically Amy replaced at the FDA. I think this is very cool. And he was on 604 early in the pandemic and wanted to do a trial to see if oral polio vaccine would produce nonspecific protection against SARS-CoV-2. I think basically what happens is it induces interferon for a while, and then that protects you against other viruses. Where were those trials done? So actually they were done by his mother in the Soviet Union many years ago. And she did trials against a number of other viruses, including influenza virus. So they were done there, and those are published, actually. You can find them. And I plan to... So Kostya has since retired, and I hope to get him onto another TWIV to talk more in depth about his career, because he really just focused on this in that episode, trying to get... He never got OPV tried for SARS-CoV-2. But the same thing was suggested for measles vaccine, an infectious attenuated vaccine. It was suggested that you could get measles vaccine and it would temporarily protect you against SARS-CoV-2 infection. And remember, this is early in the pandemic when we didn't have anything, right? Hey, I just have to highlight Noir. I love this name, Noir LeBlanc, Black the White. Welcome. And Jeff from Houston and Toronto, Costa Rica. I think I got these. I was curious, if you never had chickenpox, can you get shingles later? Well, if you never got chickenpox, if you really never got chickenpox, then you're not going to get shingles. You could get chickenpox later in life, right? And then after that, you could get shingles. But the problem is there's some level of asymptomatic chickenpox where you are actually infected, but you don't know it. There's no rash, and so it could go latent, and then you could have shingles. So I would say if you didn't have chickenpox, as far as you know, still get the shingles vaccine, because you might actually have had shingles. Okay, do you have an estimate of the number of grams of virus particles or millions of strands of DNA, RNA likely to be present in a person's body during the viremic stage of infection? Now, this is an excellent question. I don't have an estimate for that. We know how many genomes of HIV there are in an infected person. That's how we get the number. Right now, there are 10 to the 16th HIV genomes on the planet, because there are so many millions of people infected and they have so many millions of genomes in each of them. But that's all I can tell you, because grams of virus particles would be tough. I'm not aware of anyone figuring that out, but I do like this notion, because I like to express quantities of virus in something that people can get, right? So I always say, if you put all the viruses in the oceans head to tail, it would go 100 light years into space, which people can get, right? But that's really far. So I'm going to look into this, Twin Daddy. Les or anyone else who's doing stickies, please write this down and I will look it up. I think it's a really good question. Really good question. What do I think of fungus? Well, mushrooms are great. Certain mushrooms are wonderful, right? There's nothing like a nice portobello or little button mushrooms, or they're all wonderful. The poisonous ones are not so wonderful. However, there are pathogenic fungi, as you know, and I don't really like those if they're making people sick. I've never had that illness, so I'm lucky, but I like the ones you can eat and that are good. Info. FDA revised the EUAs for Paxlovid and Molnupiravir today. You don't need a positive test to get a prescription. Huh? Is that true? Well, if it's true, and I'm often responsible for accepting what people tell me and then riffing on it for a while. So I'm going to assume it's true because it's easy to check. So that's cool. So you could just go get a prescription and take it with you if you're going to Europe, for example, or vice versa. Yeah, that's cool. Let's look it up. FDA, Paxlovid, EUA. Update, because it gives you old stuff. Yeah, I'd have to read through it. Yeah, that's a good thing. You can then, presumably, you can just go get a script, right? Kip and Laura, you could just go get a script and take it with you, correct? Kang asks, are there viruses that have coats made of glycoproteins or lipoproteins? No, they're always either protein or membranes in which are inserted sugars, glycoproteins, or lipoproteins. But I'm not aware of any where the actual coat is made of those. No. By the way, today I spoke with a reporter from New York who's writing an article about the expanded oversight that has been requested to, for the NSABB for them to examine experiments that are potentially dangerous. They want to expand it to any pathogen of humans and plants. So I spoke to a reporter about it because I think this is not right, but maybe you'll see that in print soon somewhere. Do you think we're mostly done with Mpox in the States? Should the U.S. go ahead and vaccinate the general population for it as supply is available? I don't know. It's a good question. I think, look, I think we should wait and see if it comes back, and then we'll have the vaccine supply needed to vaccinate people, because I think we're very effective at stopping the outbreak in many places by a combination of information, education, and vaccination. So I would wait and see. I'm not sure that we need to do everyone at this point, but it's true that it has gone down. I actually had an Amy papers lined up about the Mpox vaccine, but we'll do it next week. PDK, how many virus receptors have genome-wide CRISPR-Cas screens been used to discover? Well, not too many, because many virus receptors were discovered before the technology came, right? So since it was discovered, some have been discovered using this, but I would say less than a dozen, because we have a lot of virus receptors already. So we identified the polio receptor in 1990, 1989-ish, and we didn't have CRISPR-Cas, and we got it. And the HIV receptor was discovered in the 80s, the rhinovirus receptor. So many of them were done before that technology. But yeah, you could use it today to do it. You could do a knockout, a library knockout of cells and see which ones are lacking it and go after the gene that way. Yeah, it can be done. So the vast majority have not been done with that. Let's see. ChatGPT. Viroids can affect bacteria by a process known as transduction, where phage carries the viroid RNA from one bacterium to another. I believe that was one of my answers, Peter, if you remember. I said, could it be brought in by a phage? And so ChatGPT is just mimicking me. But bacteria also take up DNA without a phage vector. Yeah. Not too many bacterial viroids are known. It's interesting. Let's look it up. Bacterial viroid. Maybe because I'm biased towards eukaryote. Yeah, see, Wikipedia says all known viroids are of plants, and that's not true anymore. So I don't know if ChatGPT is making stuff up. They could, right? Because that paper we did is a bioinformatic paper. They don't have the actual viroids replicating. They just have a sequence in some metatranscriptome. So, and they did find a lot that seemed to be associated with prokaryotes, but whether they're actually infecting them in the mechanism, I think isn't known. So I suspect that transduction, yes, it could be it, but I don't know if there's evidence for that. If the bladder has many ACE receptors, can bladder cancers come from multiple COVID infections? I know an increase in diagnosis is probably due to delayed diagnosis. All right. I doubt it. There's no evidence that SARS-CoV-2 infection is carcinogenic. So just because the particular tissue has a lot of receptors doesn't mean, first, that the virus is going to reproduce there at all, because there are many other steps in infection, right, that are needed for virus reproduction. But also to be carcinogenic, right, a cell has to live in order to become transformed and turn into a tumor. It has to keep dividing. And SARS-CoV-2 kills cells. So it's not only is there no evidence for that, but it's not experimentally compatible. So I would say not likely. Not likely. I'm curious about how many people who audit your lectures Know or have a background in genetics, biochem, or molecular biology? I don't know. I mean, you mean like on YouTube, people who watch? I have no idea. I suspect a lot of them have none. They're curious and want to know more. So, I mean, my students all in the class actually need a biology prerequisite, but you know, there are many, many tens of thousands of, actually hundreds of thousands of views of those virology lectures on YouTube. So I'm sure it's all over the place. And I think people have trouble. I did a live stream version of the course, and a lot of people had trouble with it because they don't have the background, right? So I wouldn't be surprised if if many people don't have the background that you're saying at all. But I don't have numbers. No, absolutely not. Can a very strong innate mucosal immune system protect you from getting diseases from most respiratory viruses? So I think an innate response is important. And I think I know one of the biggest risk factors for developing severe COVID is to have I'll call it a weak innate immune response. So there are some individuals who are genetically have changes in their genome that make them have a poor innate immune response, and so they're likely to get more severe COVID. So I think a good innate response will help in most respiratory infections. Now, remember, viruses antagonize innate responses as well. SARS-CoV-2 is a really good antagonist of interferon, and many other viruses do as well. So, the extent of protection is hard to know. I suspect that most infections of any kind stop long before anything happens, and they're stopped by the innate immune response. That's what we think. It's a very early response, and if you only have, say, a few virus particles coming in, they can easily be neutralized. If you have a few virus particles in that set up a few infected cells, that can easily be taken care of by an innate response. So, we get assaulted all the time by viruses, and I think many of them never go anywhere because of the innate immune response. So I suspect that, yes, a good innate response is helping a great deal. And I think that's how OPV non-specifically protects you against other virus infections, as we were discussing earlier. Yep. So, Ian, thank you for your contribution to science education. Apropos immune debt, why would getting infected a year later be more pathogenic? Or how does one know that getting infected now will be better than next season? So this is a good question, right? Why should you? It's a population thing, right? You have a lot of people who are in their lockdown and they're not developing their immunity, and that includes maybe people who never saw the virus before, who would have seen it that first time. So it could have been pathogenic that first time if they had not seen the virus. Because as you know, when we have less immunity to a pathogen, the infection can be more severe. And now we're seeing milder COVID because many people are immune. So in a situation where on a population level, we're not getting that immunity, the next year you're going to have, I would say, not necessarily severe, but simply more extensive disease. The severe, I think, is difficult. It may be a case of, in 1,000 infections, one is severe. So then if you have 1,000,000, you have more severe disease, right? It's just amplified because you have more infections. I don't know why on a mechanistic level it would be more pathogenic according to your logic. Yeah, that makes perfect sense to me. 400 people, that's good. That's what we left off with Q&A. And we have 300 likes, so please like the episode. And then we can get more people here. PDK says, wondering if an AV vector, and by the way, this is 2-pan virus here on Peak's icon. It's a giant virus that encodes everything but the ribosome. If an AAV vector encoding antiviral protein could be used for long-term protection against viruses. Well, what is the antiviral protein that you have in mind? I think that would make a difference. If you wanted to make an antibody, then I don't think you need to do that. I think you just treat people with the antibody. Now, treating people with other proteins is difficult. Antibodies are an exception, but therapeutic proteins are difficult. So it could be you want to deliver them in a vector. So in theory, yes. Now remember, the AAV vector is going to give you long-term production. And if that's not good, you shouldn't use it. It's not a transient thing. You know, it cures blindness because it's making the protein, you know, the receptor in the retina forever. So you have to decide whether that's what you would like or if you need a transient one. But in theory, yes, you could use it to deliver a therapeutic protein. Why not? Yeah. Julie Young, there's 373. TWIV 373. Yeah. He was a good one. Yep. Kalamazoo, Michigan. Right. Dave's Michigan basement. Love it. Yeah, I also interviewed Peter Salk, one of Jonas Salk's sons, and that was a good episode you should listen to. It's not, you know, it's not a book, it's different, but he was sitting in my office talking to me. It was fun, right by the wall of polio. Hey, this is funny. Imagine if real office hours had about 200 people waiting to see you. I have to tell my class next week that in the Earth's office hours, we had over 400 people. Yeah, we would never get that done at real offices. Well, we don't get we don't get all of your questions answered either because there isn't enough time. But you know, hopefully over time we can get to it. But in my Columbia office hours, I no longer do it in person because, well, in the pandemic we didn't have it in person, and then last year when I went back to teaching in person, I had already gone to the incubator. And I wasn't going to Columbia anymore. Now I don't have an office at Columbia, so I can't even have office hours there. So I do it on Zoom. A few students have come to the incubator, but Zoom is easy because they can pop in and out, and they don't have to go anywhere. That's fine. That's funny, Tom. Thank you for bringing this up. This is really good. When do you think it will be too late to get a flu vaccine? Well, the flu season is going to last for a couple of months. And if you assume that sorry, I was looking at another question. If you assume that protection lasts a couple of months and we're going to go till April-ish, you could get it now. So maybe till the end of February. But of course, if you're going to travel somewhere else where it's winter, it could still be useful. But anyway, for the east, for the northeast here, I would say you could still get it now. Well, here's a bacterial question, which I shouldn't answer because I'm not a microbiologist. Maybe we should get Michael Schmidt here one night to do office hours. Then you could ask lots of bacteria questions. Anyway, he's just a generally entertaining guy. And he would do it. Is that a good thing, folks? We want Michael Schmidt? Anyway, why is it not contagious? Because it's not multiplying. It's in deep in your lung in little nodes that are not multiplying. So there's no bacteria to come out of your lung and infect others. That's what I would say. But We'll have to ask Michael when he comes on. What about vitamin D from sunlight and immunity? Well, vitamin D is a contributor for sure, but most of the data is anecdotal, and really good clinical trials are hard to do for that. So I would say at the beginning of the pandemic, everyone said, take vitamin D. Actually, I had been taking vitamin D for a long time because multiple physicians said, look, you don't get a lot of sun in the Northeast, especially in the winter. Just take vitamin D to compensate for it, because that's what the sun is doing. So, it doesn't hurt. That's the good thing. And Tony Fauci took a lot of vitamin D, right? It's not going to hurt. John says, it would be great if you could cover livestock viruses on TWIV. Sure, I think that's a good idea. And we have a veterinarian now, right? We have Angela who can do that. By the way, John, I met at UConn, University of Connecticut stores when I went to interview, I can't remember his name now, Philip Marcus, who was was the first person to clone HeLa cells. That's another good episode, that one, because he told his story. It was very cool. Speaking of polio, for those of us who got IPV in the 50s, do we acquire gut immunity when exposed to the circulating vaccine poliovirus? Yeah, you will get infected, you will shed virus, and you will mount a mucosal immune response. But like everything else, it's transient, right? So your antibody and T cell levels will decline after so many months, so your gut will then be susceptible again. But you will have immunity. You'll have immunity, and so that if the next time you're challenged in the gut, you will have a memory response after a few days of reproduction. Remember, it doesn't initially inhibit infection because there's no antibody there. You have to wait for the memory response, which is going to be a few days. But yes, you will get infected. You will get some kind of immunity. All right, so this is a question that I asked Daniela Weisskopf a couple of episodes of TWIV ago. Okay? And I said, how do you know how long the T cell responses are lasting? She said, we really can't answer that because, if you get someone, so you vaccinate someone and you send them out there, they're going to get infected maybe every year. So you can't tell how that initial vaccine course does with durable T cell immunity. You just can't do it. So, the first SARS-1, you can find people 17 years after that outbreak with T cells against the virus. Whether they're protective against severe disease, we have no idea. And so maybe it would last that long, but we just don't know. And she said, and she's a T cell expert, I'm just quoting her, can't know how long it's going to last because you have a combination of vaccination and infections all the time. I suspect that we're just going to have mild disease. Everyone who's been vaccinated and then infected on a regular basis is just going to have mild disease going forward. And if they have risk factors or if they're over 75, take Paxlovid. And that's going to handle most of the issues, not all of them. It's going to handle many issues. The Discord is nice. Barb Mack is a moderator there. So is Peak Dunning-Kruger, and Les is going to be a moderator there as well. It's a good little environment. It's kind of like this, except there's no me talking. There's just a bunch of people talking, and it happens 24/7. Yeah. So Charlotte took the flu shot in September. Should I get the flu shot again in February? I was looking at this on the CDC site, and they do not recommend 2 flu vaccines in one season. Now, tell me if I'm remembering, did Daniel say it was okay? I don't remember if he said that. You could take one in September and one in February, but this is not recommended by the CDC. I don't know why. I was trying to find that today, but I didn't know why. So I would not do it because of that reason. Well, Mike was vaccinated with a cutter batch of Salk vaccine. Yeah, you're lucky you didn't get it. It was a few hundred kids who got it out of the tens of thousands of doses that were given out. So it's good that you didn't, right? Is there research going on to determine whether fourth or fifth boosters increase protection from severe illness or death? Yes, there is. But in my opinion, it is flawed. In one of the papers I had up today, go to the Amy papers. There is a paper there that's relevant. Here, I'll bring it up. Let me bring up the page. Okay, where are the Amy paper? Oh, I have to type it in. The Amy papers. Here we go. Let's turn on my sharing here. Where's the sharing? Screen share. Here we go. Where is it? Here, the top one there. Effectiveness of bivalent boosters against severe Omicron infection. So this is a study out of North Carolina, okay? And they conclude the effectiveness of these against severe disease. So what did they do? They looked for, sorry to go up and down. This is not my highlighted copy. There's COVID-related hospitalizations and deaths. That's what they're looking at. So, first of all, we have to be careful about COVID-related hospitalizations. I don't know if they did it right. They don't really give a lot of information. The data sources have been studied elsewhere. You'd have to go look at that. And then they said during this period, May to August, that these people received monovalent boosters. All right, so May to August, and 61 of them had hospitalized and 23 deaths. And then we're looking from September to November, we have people who got the bivalent booster. We had 57 hospitalizations and 17 deaths. Okay, so it's less, and they calculate the hazard ratio and so forth. The problem is it's a different period of time. This is an observational study. It is people vaccinated May to August with the original booster and then September to November with the bivalent. So it's not fair, really, to compare it because we're farther away from the May to August, so the protection is going down. And, in fact, they see protection from the bivalent going down as well. So we're all going to go down. Eventually, if you're 6 months out, you're going to have the same protection against severe disease with both, with either booster. So this doesn't tell me that the bivalent booster is any better than the original. Here's what the booster does. For 3 months, it gives you high antibodies. It protects you. And then it's over. Is that how we're going to deal with this? Well, that's what we do with flu, apparently. We give you a flu shot every year. We match it to the circulating virus. You get protection for the flu season, and then that's it. So maybe that's the COVID strategy. I'm going to ask Paul Offit tomorrow what he thinks about that, because it looks like that's where we're headed, right? Okay. Sorry to dwell on that. So, yes, there are other studies ongoing as well, and we'll certainly hear about them as well. Australia's study claims that oral antivirus within 1 day is better than 5 days. I don't know the study, so let's say this is correct because I haven't seen it, and I shouldn't comment on it until I see it. But if it's correct, I'm not surprised, right? The closer you get to onset, right? At onset, you have less virus than a few days afterwards. So it's easier to inhibit when you hit with antivirals early on when there's less virus already. So it makes sense to me. It's just hard to do that sometime, right? One day after symptom onset, people typically wait a little bit. And so it's hard to do. But I think, the closest you can get. So when I got COVID symptoms, I got on Friday. Saturday, they got worse, so I started Paxlovid on Saturday, and the next day they were gone. So I caught it pretty early, right? Day 2 after symptom onset. So I'm not surprised. I'm N=1, so it doesn't mean anything. But theoretically, I think it makes a lot of sense that you treat earlier and you're good. Have I ever worked with any BSL-4 organisms? No, I have not. I visited BSL-4 laboratories, but I have not worked with the organisms. Thank you, SRR, for your contribution. Well, I don't know anything about vinegar and Dawnazan spray, but I do thank you for your contribution to science education. If anyone has any information, please help SRR. Jeff says people are suspicious of what they don't understand. Keeping that in mind, we are at fault because our K-12 educational approach to teaching science is horrible. Well, I think you're right on both cases. That people are always suspicious. They're afraid of what they don't understand. Absolutely. And the other is, yes, our science education is not good in this country. I have no idea how you would fix that. It's as you saw from the previous administration, even the Department of Education got politicized. And so I don't know how you can change this. We have to basically give teachers a better science education to begin with. And I just don't know how you do it. I'm going to go teach at a school in Washington, D.C. in May to try and help. But, I'm only me. It's going to be, 20 kids. So, but I do think you're correct about both. Simon says anecdotal data. Life is going on normal in Shenzhen. Okay, I don't know. I have no idea. Polio Pete, hello. Are there ever new influenza mutations? Seems like they're always looking for past strains. So yes, people. Influenza viruses are always mutating, like every other virus. But the key ones are the ones that impact antibody and T cell reactivity, and so they check every year to see if a change in vaccine is warranted. So there's a huge network of laboratories around the world in both hemispheres where they're looking at circulating strains of influenza viruses and trying to figure out if they should change the vaccine. And so some years they don't and some years they do. It all depends on how well the circulating viruses react with antibodies. That's the main test that they do. So yes, it's always changing, and some years we change the vaccine in response, and other years we don't. So, the plan for COVID vaccines, I don't know what the plan is. I'm going to ask Paul tomorrow, but they said let's harmonize and get everybody on the same vaccine, maybe a bivalent. And do we need the ancestral virus in there at all? Should we change it every year? What data do we need to change it? Those are the kind of questions they're addressing. When will the H5N1 outbreak in poultry peak? I don't know. No idea. That's a good question. This is a long one. And yeah, the eggs are expensive, I know. But I don't know when it's going to peak. I have no idea. And why it's so extensive, I don't know either. If someone says they know, they're just making it up, okay? Because nobody can predict when it's over. And I don't want to hear any modeling telling me when it's going to be over, right? No, we just don't know. It's an unfortunate situation, and that's the end of it. Yeah. This is the car talk of virology. Yeah, but there's only me. Car talk was 2 people, right? I need another person. So when Amy's here, it's the car talk. But when it's just me talking. And, it doesn't have to be me every week. As I said, we'll get some guests here. We just have to find people who are willing to stay up. Well, we can do an hour with the guests from 8 to 9, right? Lise is curious the percentage of folks who have a reaction to Shingrix, super exhausted, muscle joint pain, etc. I had very little reaction to the second shot. The first, nothing. The second, some arm soreness, fever, which I controlled very easily, but not really debilitating. I did my normal treadmilling and everything was fine. Yeah. Thanks. I'm glad you like them. I slightly modify them each year. I feel I get better at delivery every year. I know what works and what doesn't, so it's worth listening to. And repetition is always good, for sure. What is a furin cleavage site? And does SARS coronavirus 1 contain one? No, SARS coronavirus 1 does not. But other coronaviruses do. And some of the common cold coronas have a furin cleavage site. Now, what is it? So we have a protein. Let's see. What can I bring up here for a protein? Can I use my charging cable? Here, this is a protein. And a furin cleavage site would be, this protein is made up of a sequence of amino acids, right? A furin cleavage site would be in here, between a couple of amino acids, and then a protease, an enzyme that recognizes it, will cut the protein in 2 pieces. That's a furin cleavage site. There are many cleavage sites for all kinds of proteases. The M protease of SARS-CoV-2 is a protease that hits a specific amino acid sequence in the SARS protein. And that is inhibited by the way, Paxlovid. So furins are relatively ubiquitous proteases throughout the body, and they cleave one of 2 sites on the SARS-CoV-2 protein. There are 2 cleavage sites on the SARS-CoV-2 glycoprotein, the spike. One is cleaved by TMPRSS2, which is another protease, and one is cleaved by furin, and both cleavages are needed to have efficient fusion of the virus with the cell so that the genome can get inside. So the furin cleavage site, of course, was the focus of early attention because SARS-1 didn't have it and SARS-2 did. So, it must have been introduced by people. I mean, where do you get that logic from? Just because it's in this and not in the other, you say it's introduced? Keep your mouth shut until you think about it. Oh, my gosh. Other coronaviruses have it, and that's what it does. It's needed for efficient reproduction of the virus, for sure. It's worth noting that John Udell on Twitter had the guts to call out the left is just at fault as the right, and they lie just as frequently. I don't know if they lie. Maybe they do. I don't know. I don't know what you're talking about. To call out the left for what? But certainly both sides have issues. But, one side says, don't wear a mask, don't get vaccinated in some states, and that's not right. Any thoughts about the bourbon virus? The bourbon virus was discovered quite a while ago. It's a tick-borne virus, and discovered in some farmers, or a related virus was discovered in farmers. I'm probably thinking of another virus related to it. Bourbon. You type bourbon without virus, you get very interesting hits. It's a tick virus first found in Bourbon County, Kansas. There haven't been so many infections in the U.S. Yeah, it can be a problem. Fever, tiredness, rash, headache, body aches, nausea, vomiting. So why haven't we known it? Why don't we? Isn't it widely known? Because there aren't so many infections, and that is the key with anything. If it's not fatal, and if there's not a lot of them, nobody cares. But it's out there, and we talked about it on TWIV. It was actually on an old episode. Let's see if we can find TWIV bourbon. Would that get it? Yeah, bourbon virus. Yeah. TWIV 322. That is in February 2015. Postcards from the edge of the membrane. The TWIVodes answer questions about bourbon virus, etc. Yeah, so we've talked about it, and probably subsequent to that as well. Should get Mark Veldone on. He's been doing a great job communicating. Many people are doing good communicating, and ours is to do podcasts, and everyone has a different way of doing it, and some do it well. And I think this is what we need. We need to have a whole range of scientists doing this kind of communication. I have a question. I am an ecologist at UT Austin. I attended a lecture by a member of the Warby Lab. They claimed there was no evidence of original sin. Is that still true? So, John Udell said, what is this original sin? Why are we calling this? Because it's a natural feature of the immune system. You get infected or vaccinated with something, then you're vaccinated with something related to it, and you preferentially get an immune response to what you were originally vaccinated or infected with. That is the way the immune system works, to put it in Udellism. And if the second exposure is different enough, then you don't have that. So people discovered this, and they called it original antigenic sin. And now, to get away from the religious reference, they call it imprinting. But it's the same phenomenon. And yes, it happens with SARS-CoV-2. If you are vaccinated against ancestral virus, and then you get the booster, the bivalent booster that has some Omicron in it, you preferentially respond to the original because they're very closely related. And yes, that's imprinting. So it still happens, but that's the way the immune response works. And it's good. How about Columbia's Miller Theater? Can you write that down, Les? Columbia's Miller Theater. This is a good idea. Maybe they would make it available to me, and that would be not a bad location. We could take the one train up there. Yeah, and it's big, right? Columbia Miller Theater, Broadway and 116th. Venue booking. Stop with the cookies. Plan your event. 668 seats. Everybody going to come to TWIV 1000. Oh, that's a good idea. Thank you. Thank you very much. Yeah, that's a good idea. If you're interested, send me an email. Vincent@microbe.tv, and put 1000 in the subject line. Because if we can get a 668-seat theater, hey, everybody can come. Those are all the people in the world that would come in person for TWIV 1000, right? Wow, cool. Is there a way to erase the imprinting? No, you can't, because you now have B cells that are reactive with the original antigen. And anything that cross-reacts is going to tickle those B cells rather than make new B cells, right? All right, so how could you get around it? Make a new antigen that doesn't cross-react, but still will give you protection against the pathogen. Not so easy, right? Because you presumably got that the first time. But that's how you would do it. If criticism of the bivalent vax is that we're chasing variants that will change. Why isn't this also true of the flu vaccine? It is, in fact. But we don't know as much about SARS-CoV-2, and it's not clear to me that you need a new vaccine to provide protection against severe disease. Even with extensive antigenic variation, it may be sufficient to have the original vaccine. And we don't have enough data, I think, on that. And that's what Paul Offit always says. Show me that a variant-matched vaccine is any better than the ancestral. It hasn't been done yet. But what I think happens with the flu vaccine is you get a flu shot and you get protection for 3 months because your antibody levels are high. And maybe T cells as well. And then it goes away, and then that's the end of the flu season. And then the next year you get another one. So is that how we're going to deal with COVID? That might work, but I don't know that you have to change it every year. We need to have data saying that you could use the same vaccine for COVID every year, or you need to match it. So far, I'm not convinced that you need to match. And so why go through the expense of updating it every year if you don't have to? That's the key question. Does it need to match? Actually, do you need to do it every year? Because I think it's for people at enhanced risk. Because young people, that vaccine will protect them against severe disease with no other health issues. How long? I don't know. We have to do those studies. And maybe we get some information on whether you need to boost or not from that. But just don't go ahead and do it without the data. That's what Paul Offit is trying to say. Don't make policy without scientific data. And I think that's fair. Oh, here's another thing. Jack Dixon's nephew's jazz CD. So I already said this. They're going to play at TWIV 1000. Sorry. Are there any microbes virologists are searching for that haven't been discovered yet, akin to when scientists first discovered filterable viruses? Well, if you don't know it's there, how can you search for it, right? Usually you stumble onto these things. So I would say, no, not that I know of. I mean, we're trying to solve mysteries, right? Yeah, where did life begin on Earth? How did it begin? That sort of mystery. And maybe in the process we discover some other things. But, you keep looking. You find new viroids that infect other organisms that you didn't know about. And so you just keep looking with some goal in mind, and you can find other things. But no, I'm not aware of any microbes being sought. But, over on TWIM, we talk about people finding a microbe that's huge in some funny environment where people are just searching, and they find the biggest bacteria ever, right? And really interesting features of it. So you just keep looking, and you're going to find it. All right, so to be clear, OPV does not prevent transmission. Would it at least reduce the extent of transmission due to production of IgA? So when you first get it, you will have high antibodies in the gut, correct? And for, 3 months, you will impact transmission. Then those antibodies will have gone away. They do not last. Secretory IgA does not last in the gut. So then if you are infected with poliovirus, there'll be a few days of reproduction before the memory response kicks in. And then that will so I would say OPV inoculated person might have limited transmission. Now, we can't test this. We haven't tested it. It's a very hard experiment to do now because there's so little polio left and you can't challenge people. But that's the theory, that you still are going to shed even after OPV. Certainly after IPV, there's no question about that, but OPV as well. Okay? That's where I think it stands. So Pete would rather see tulips than TWIV 1000. That's fine. You can make your choices. No problem. Dixon's Nephew's Band should play that piece that was written for the Richard Ernst Award. Right. I have to get it. The guy hasn't sent it to me. I asked him to send it. Oh, no, he did. I do have the music, right? It's written for 4 French horns. That's a good idea. So I'll give it to him. And maybe they could make it work for saxophones. Yeah. Ooh, look who's here. Cam. Hello, Cam. How are you? Good to see you. Apparently, when under a year, I had 1 chickenpox blister on my rear. Okay, so you had chickenpox. All right, do you know of any good intro immunology book or course? Take Brienne's immunology course. It's on YouTube. You could find by searching her, and probably Les will put a link in shortly. And I'm sure she has a recommended textbook for it. I don't have anything that would be suitably introductory for you, no. But she has one recommended. So I would say get her her lectures on YouTube and see what she's recommending. What's the big thing we don't understand yet on polio? If you could give unlimited grant money to study basic polio virology, what would you finance? I think we should figure out this cross-reactive story that Amy is investigating. She discovered that antibodies to polio can cross-react with other enteroviruses and vice versa. And I think that needs extensive investigation to find out its significance in terms of pathogenesis. So I think that would be a main one that we should look at. You realize that Amazon is dropping Smiles. Yeah, I do. So I'm just ordering everything on Smiles now until then. It's too bad. Here's a great post from Randall Munro about the total amount of viruses in the world. Okay, that's cool. Like that. You can't, regular folk can't post links. The mods have to post links. So if you can tell them about it, they will check it out and post it for you. So Vanity finds some copies of A History of Polio that are not too expensive. So that's one other option is to look around at used bookstores, yeah. One reason I've heard for why there is no and can be no HIV vaccine is that the virus changes rapidly, but flu changes rapidly. What's the difference? Well, I think the main difference is that a vaccine for HIV would have to be sterilizing, and no vaccine is sterilizing. So HIV will establish a latent reservoir, right? And therefore you can't prevent it. So when you get infected, within 2 weeks, you establish a latent reservoir where the viral genome integrates into cells. You would have to stop every virus from getting into cells in order to do that, and no vaccine can do it. We haven't figured out how to do that, and that's really the issue, for sure. How does OPV cause polio in the unvaccinated? So when people are given OPV, you take it by mouth, goes into your intestine, reproduces, and during the reproduction, it reverts to neurovirulence. So all the mutations, or many of the mutations that make the virus safe as a vaccine, go away, and you shed revertant virus that's now able to cause polio. That circulates extensively. So if you're vaccinated, it doesn't cause polio in you, but if you are unvaccinated and you encounter it, it will, because it's no longer a vaccine. It is a revertant. And that happens in every kid that gets OPV. The vaccines revert. And in 1.5 million recipients of OPV, the kid itself gets paralyzed from the reversion. I saw an anti-vaxxer say all of their cells are mRNA-free. Okay. No, they're obviously not, because you need mRNA to live. Do malaria-carrying mosquitoes live outside of Southeast Africa? Yeah, absolutely. And other countries as well, for sure. Asian countries. Not Florida, as far as I know. They're Anopheline mosquitoes. Let's see their range. If they are. Most of the continental U.S. has Anopheline mosquitoes. You know, there were at a time, there was at a time yellow fever in the U.S., but that's a different mosquito, of course. But there is no malaria unless it's imported into the U.S. But that's a TWIP question. We have to get Daniel Griffin on for that. For between viruses and bacteria, are there estimates of which cause more disease? That's a good question. If you look at the top causes of disease, well, the number one, of course, is cardiac disease. And the, one of the ones up there are infectious respiratory diseases. So I don't know. It's a good question. I think that's one I haven't come across. And it's not something that's going to be Googleable. Numbers. Are virus or viral infections are more common than bacterial infections? But I don't know. And I like that. And that's something that needs to be looked at. Malaria-carrying mosquitoes were a huge problem for settlers in the southern colonies during the initial settlement of North America by the British. And that's from Peak, our moderator. Thank you. You can tell Peak is going to school because he knows things. Ted wants to know if viruses mutate within humans. And if so, can someone with a virus in their system become infected by a new variant within their own system? So, yes, they do mutate within us. If the infection is short, the consequences for the host are minimal. So a 4, 5, 6 day infection, that kind of mutation isn't going to make much difference to that host. Maybe another host. But for long-term infections, hepatitis C virus, HIV-1, yes, the mutants that arise in the host have consequences for the host. So for Hep C, mutants arise that are resistant to T-cell killing, for example, and that's bad for the host. And in HIV, mutants arise of all sorts of different properties, one of which is resistance to antibodies, but others receptor utilization and so forth. So yes, within a long-term infection, it makes a difference, less so within a short-term infection. Are there any amateur virology societies? That's a good question. Amateur virology society. We have professional virology societies that I am aware of, but I don't know of any amateur virology societies. Maybe someone needs to start one, but we have Q&A, we have office hours, and now we have Discord. So that should be enough, right, to get your virology fix. Tim says, if chicken farming stopped and we could grow chicken meat in the lab, could that greatly reduce the number of new flu strains or pandemics? Well, yeah. That's not enough, because chickens are not the only host, right? There are all kinds of wild birds, particularly water birds, that are the natural reservoir of influenza virus. And then, of course, we have turkeys and geese. So, no, it wouldn't make a difference in terms of influenza virus anyway. Peak says, yeah, we have an amateur virology site. It's called the Microbe TV Discord server. Yep. Are there any vaccines that actually target antigen to dendritic cells? And would that be better? For example, by targeting something on the surface of DCs. Yeah, you could somehow incorporate into the antigen something that would bind DC sign right on the surface of DCs. But no, I don't know of any that do that. So you could do that, and you could also deliver the vaccine to the skin where there are lots of dendritic cells, right? So Carl says, amateur virology is called the comments section. It's good. Anyway, Tim, it's a good idea. TLM. Eric says there are 11.9 micrograms of virion produced per day in an actively infected patient. 11 times 10 to the minus 16th grams per HIV virion, and that's the number produced per day. Thank you, Eric. My partner's a sound engineer. If we were in New York, he may do your sound. Well, let me know. I need to know soon because otherwise I need to engage someone. From Virology Lesson 2, for plaque-forming units, is it assumed that one susceptible permissive virion results in one PFU? No, it is. Well, what's assumed is that one particle is sufficient to cause a plaque, right? But not every particle is infectious. So 1.7 times 10 to the 8th is PFU per mil, not virus particles per mil. You could have 1.7 times 10 to the 9th particles per mil if 1 in 10 are infectious, or 10 to the 10th if 1 in 100 are infectious, okay? So one particle is sufficient to initiate a plaque, but not all particles can. Good question. Why is canine parvovirus so hard to fight? Well, it's my understanding that the vaccine is effective, right? But I don't know other than that. Maybe we have to get Angela to answer that. Let's see. Do you think the SARS-CoV-2 variants can now be defined as strains? It depends which ones. They have to have a different biological property. And I don't think that immune evasion is sufficient to do that. So we have to decide what property is different enough. Thank you, John, for your contribution. Really appreciate it. How long will my puppy be ready after vaccination to fight disease? I think it's a similar response as humans. I would give it more than 2 weeks, but, a month should be sufficient. Oh, Dave was a biochem major at Purdue, dropped out for an apprenticeship as an electrician. Well, you probably made a lot more money as an electrician than you would have as a biochemist. So good for you. So what's the action mechanism of the shingles vaccine? Since immunity and antibodies to varicella are already present. Well, when the virus reactivates from neurons, it antagonizes immune responses, and having the enhanced immune response conferred by Shingrix helps to control that infection, which wouldn't normally be controlled by immunity. And if you're interested in to have 1,000, send an email to vincent@microbe.tv with 1,000 in the subject role, subject part there, and if we have room, we'll let you come. But if there's not room, we'll do some kind of a prioritization. Thank you, Aaron, for your contribution. You have a good 1918 flu book. Well, we just interviewed Jeff Taubenberger, which was really good about 1918. But Gina Colada has a good book about it. Of course, John Barry has a good book about it, which is not free of errors, but it's got the sociological parts of 19 influenza down. It's good. Any update about Novavax for COVID? My understanding is that they're going to FDA with some other uses as boosters and combinations, and so we should be hearing about that. I don't know anything about it. So Eric took Philip Marcus's virology course. Great lecturer. Cool. Very cool. Yes, John, we met in 2012. Exactly. Vanity says, who wrote the book, Vanity Nutrition, people who regularly take vitamins usually practice other healthy habits, eat healthfully, exercise, get rest regularly, wash hands. It makes sense that they'd get sick less frequently. Okay. I skip over some questions that I feel I am not capable of answering, and I don't want to spend the time to say I can't answer that. Don't take it personally. Why do some people experience fever with viral infection and others don't? It's a good question. It's genetic basis. Some people are genetically predisposed to responding with the cytokines that produce fever or to detect the pathogens, and others are not. I'm not aware that we actually know the mechanism. We do know that fever happens when pathogens are detected by our innate immune system. We make cytokines that then crank our fever up, our temperature up. And so there are differences in sensing and in making the cytokines. Everybody's different genetically. People are not the same, so it's not surprising that happens. Daniel did say he takes 2 flu shots separated by several months. However, this was not done. Yeah, the CDC guidance is not to do it. So, all right, I'm going to start wrapping up here because soon I need to do TWIV with Daniel Griffin, and I need a few minutes in between to get set up. So let me just scan through the yes, the bivalent study stopped after 99 days, simply short-term antibody boost. That's the problem I have with a lot of these studies. You're looking at short-term antibody boost. You have no idea whether there's any advantage over the old vaccine beyond that short-term antibody boost. It's not really a fair comparison. $422 people. That's great. I love it. And come back next week. I know I'm skipping over a lot of questions. What's a Discord server? There's the address. It's a website that you can go to. You just sign up, and it is a place to talk to people. You can ask questions. People will help you. Absolutely. I am looking for one particular email here. When I finally publish a paper, will you review it on TWIV? Hey, man, if it's good, yeah. I received an email that TWIV 280 was posted. It's there, isn't it? Let's see. Oh, let's see. I didn't post it. My gosh, I'm so far behind. You're right. I haven't posted it yet. It's only 279. I'm so behind. I have to do TWIP. It's in a can for a week. Oh, my gosh. I'm so sorry. And tonight it's not happening because I have another hour with Daniel. I'm looking for one email from a former student who took my course. Maybe I'll get to it as I scroll down here to the bottom. Missing all these good comments. I'm so sorry. Come back next week with them, please. I'll have to search for it. There is a search function here. But first I will scroll down and thank anyone who has donated here. Okay, here we go. Mr. Ozzycamp, thank you for your contribution. Good to see you again. Thank you so much for supporting science education. John, thank you. Lots of great questions for the first office hours. I felt like I knew the answers to some of them, too, because of the free lectures. Go check it out if you haven't. Thank you, John. Really appreciate that support. Now, let me search for... Here we go. Here is Alexis. I took your class last spring while I was a combat medic in Ukraine. I remember this because Alexis, partway through, she said, I have to go to Ukraine. I'm going to try and keep up with the class. And then her car got blown up. Oh, my gosh. You're so brave. I tell everyone how wonderful your class is. Thank you for all you do. Best class at Columbia. See, a lot of the students say that. Best class at Columbia. Thanks, Alexis, and stay safe. Good to see you again. All right, I'm going to wrap up this episode of Office Hours. I want to thank all the moderators tonight for helping out. We'll go to full 2 hours next week, but I have to, as I said, hop on with Daniel Griffin and do the clinical update. We had Steph, we had Les, we have Tom, we have Vanity, we had Frank, we had Barb and Peak Dunning-Kruger tonight. We had 400 plus of you. And look at this, we have as many likes as people here. That's a real first, if anything. Thanks, everybody, for coming. Your questions are all fabulous. You're all fabulous. Come back next week, same time, 8 p.m. Eastern. Meanwhile, of course, stay safe. Thanks, everybody, and good night.